So de novo always assumes there's something in another protein for which you need a lookup. It's somehow you need something to know about that. It may not be protein A that you need to know something about, but there is a set of proteins for which you somehow need to know something experimentally. Ab initio, in principle, you don't need anything. That's the idea. And again, this hardly ever exists. This is sort of the big difference here is that de novo implies, for instance, machine learning extracts principles from 10 different proteins. For each of those, we know something about, for instance, the sub-cellular localization. It does not then infer sub-cellular localization based on one of them. It somehow has sort of an aggregate truth to those 10. While by homology really uses that one, but is assumed to be evolutionary related, okay? And in these three different, or in two of those, I'm going to predict, or I'm going to show you methods that predict sub-cellular localization. Again, this is Mycoplasma genitalium. In bacteria, there are not many localizations, but there is a densely packed cell. This is what this image here is supposed to give you. Bacteria essentially have a membrane. They have something inside, plasmid, ribosomes, so there's some substructure, so to speak. The flagellum propels it, but essentially it is a wall, and inside and outside. This is an oversimplification, but one that has some validity to it. While the eukaryotic cell has much more structure, substructure. So DNA processing is done in the nucleus, and there are many other compartments that are really specialized for certain purposes. Some of these compartments, such as these mitochondria, for instance, are thought to have been taken from bacteria. So essentially the idea is that at some point long ago, a eukaryote ate essentially a bacterium that became the mitochondria. Now, interestingly, mitochondria have a genome. This genome currently codes, so the typical bacterium has about 1,000 proteins, 1,000 to 4,000, right? And so mitochondria have about 2,000 proteins. So the whole story fits, right? However, of the 2,000 proteins that are today in the mitochondria, none of them comes from the bacterium. So essentially the bacterial genome inside of the mitochondria has completely been replaced over millennia. So there is a historical connection, but it's not really today, it's not like you have a bacterium inside of you. You all know that we have bacteria inside of us. We are full of those, and they are important for the way we function, but not inside of a cell. Okay, now the simplest task, as I said, is to predict outside, inside the cell, and nuclear, in these three states. So how can we predict sub-cellular localization? Oh. Yes? Sorry.
Last time I asked a question and you asked me to ask it today.
Yes.
When you, well, I wrote it down like this, why does it even work to predict sub-cellular localization when all homolog proteins are removed from the data?
Okay, so we are getting back in some sense to these words here. So when we predict by homology and we remove all homologs from the data, it does not work.
Okay.
Full stop. The homology-based inference means exactly the inference for that particular protein that is homolog. If I remove that because of a bias reduction or whatever other reason, that method never works. De novo somehow tries to sort of, de novo typically is, for instance, machine learning. You learn some principles, some statistics. You said statistics, machine learning, simple statistics could be amino acid composition or something like that. And you relate that to sub-cellular localization. Then it is, and then you remove any, so the one you want to predict, you remove anyone that is really similar, but it still is somehow similar on the level of there is a protein that in machine learning by statistical relation you can recognize. Okay? So homology-based inference, the moment I remove the homolog, no longer works. There could be a more distant homolog, a second homolog, a third, a fourth, but if you remove all homologs, it's no longer working. That's a very important point. Now, this is exactly this point, so if I infer by sequence similarity, if I remove the sequence similarity, if I remove everything that is sequence similar, it never works. Text similarity we will talk about, motifs we will talk about, and structure and sequence-based de novo we will talk about. So the first is the homology, and this is exactly this point. So in order to find out how well homology-based inference works, and we have done this, I have shown you that before, you take all proteins of known localization, experimentally known, take a subset of sequence unique and do this against all. And now that was the point to exactly your question, Nicholas. If you had done unique against unique, so there is no pair in there that is recognizably homolog, you wouldn't find anything. Okay? So that's why you have to somehow keep this bias here. So the large blue has more bias than the tiny blue here, because the bias has been removed by design. And best would be to do biased, non-biased against non-biased, but then I remove the signal. Okay, that's why I cannot do that. And the next best thing could be to sort of somehow remove the bias. But that is getting tricky, so we do unique against the whole thing. Again, you could argue. And again, so this is one particular record out of the SWISS-PROT database that annotates the function about 500,000 proteins, is expert-based, experts who read literature, and there is one particular keyword, sub-cellular location, and in this particular case it's nuclear. I showed you that it works, the blue one here, in an E value, in an H value, to some extent very similar ones. Here you can do it at levels above 80, and then you hit some point where you lose the signal. Now, text similarity, how can we do that? The idea simply is that is work from Rajesh Nair. So say you had that SWISS-PROT record, but I removed the word nuclear. I removed the column sub-cellular location. And for people who know a little bit about proteins, they will then read here and will see that this is a transcription factor. And transcription factor has to do with the protein that's being turned on. That means this processing of DNA in a eukaryotic cell. Essentially most DNA and processing of DNA is done in the nucleus. So just reading that and knowing the background information, if you are an expert, you can infer this keyword even if it's not there. So now the question is, can we do that automatically? Can we teach a machine to do that? And so one thing is, can we in fact do the trivial ones? It binds DNA, so it's nuclear. It has to do with block coagulation, so it has to be outside of the cell. So those are trivial for experts. Can machines learn that? But in fact, ideally, we would be able to even find non-trivial ones. So those that are not such that an expert could immediately say DNA binding, okay, that's nuclear. Maybe there are keywords that we can deduce, or there is information in there where an expert cannot immediately say, okay, this is nuclear, this is extracellular, but the machine combines it all and gives a probabilistic prediction. The way to do that is, well, again, we begin with a data set of sub-cellular localizations that are annotated. From all of those, in this particular case there were 15,000, we extract all the keywords like this. So now how would you do it? How would you develop a method? How would you turn this idea, this simple idea, into a method? Essentially, very clear, you want to put all the keywords that are relevant to recognize sub-cellular localization. Yeah?
This looks like a job for a neural network.
Neural network is, yes, so let's just stay there. Let's stay at the point of the neural network. So the first question there would become, what's your input?
The set of, like, you have a vector, where each element of the vector represents one of your keywords, and you say either yes or no.
So you take all the keywords in the entire SWISS-PROT.
If
15,
it's not too large.
Well, you begin to see that, right? So we have 15K of these. And you're already 15K times the number, roughly the number of words you see. I do not know how many words we see. My rough guess is it's ballpark 100, right? Yeah?
But then you can take a subset of all the keywords.
How do you do that?
we take some experts and put some knowledge, initial knowledge in and say, okay, we choose.
I'm not sure whether you were the one who's
50
but yes,
because they are really like transcription vector. I don't know.
So
put in those, and then you can.
Let me look at all of you. Do you know a biologist? Do you have your favorite toy or token biologist who you can get to work for you to annotate that? No. Not? Okay, that's not the solution. We have to find another way. They're not easy to get by, these. Expertise is something that you learn after a long time in the study. Yeah?
Okay, we assume that this takes a long time for many proteins, but if we take a small subset and see which keywords...
It's not necessarily about... okay.
Which keywords make a difference?
How do you do that? Can somebody else fill in? Can somebody maybe take his ball? Yes, a subset.
To reduce the complexity.
Yes, but how can you find it? How could you get a subset?
You could analyze, I remember from text analysis, what was it called?
I mean,
IDF, so you can weight the words, whether they occur very often. If they occur in every function list, then they...
Exactly. So that, for instance, here, right?
Yes,
it's totally nonsense. So you will, in the simplest, you immediately, Matthias already defined the simplest way in which you can do it. You can say, okay, I want to have keywords that define my subcellular localization. If I have words that you see in every single subcellular localization, they're already gunners. They have no entropy, they have no information content. And entropy is essentially already the solution. And in principle, this is exactly what Rajesh did. So he listed all the keywords that are informative. So this is a list of all keywords. It's of course continuous. Does it for, in this particular case here, 3 different subcellular localizations. So this is one protein. One means simply this keyword has been observed there. So 1 and 0. Keyword is there or not there. And this again is a very, very, very long keyword in principle. And then that creates an experimental database. That experimental database we have to filter. We have to kick out things that are not informative. Bring it down to something. But the moment we have these vectors and assume that the keywords we have are somehow informative. So informative now, remember, means 2 things. One is, it's not in every single one. It's not total nonsense. But it is also not a keyword that is only in one. Because those are the trivial ones. We also want to learn non-trivial ones. So we do want to have keywords that are observed in more than one subcellular localization. And somehow, by the force of the vector, we can assign, right? So we kick out one extreme, all subcellular localizations. But we do not kick out the other extreme. Meaning, or we do not only keep the other extreme, which is only in one subcellular localization. That is not what we want. We also want to do that. This would essentially sort of reproduce in some sense the expertise of a biologist, or some of it. But we do want the correlation of overlap, okay? But now comes the next question. Assume I had this database, right? I gave you 1,000 keywords. We are still pre-neural network here. We could possibly do that with something like that. But let's think about something even simpler at this point. So I gave you say 1,000 keywords that are somehow informative and relevant. A database. So for every single one of these keywords, I have an assignment, whether it's in the nucleus or not. Now you come with your new protein. Okay. How do you determine in which subcellular localization it is? Again, that could be machine learning. Yeah.
Just sum the values over the vector and see which one is nearest.
Yes, but now we have an issue. The issue is that that if you did that, what you implicitly assume is that the annotation you find is complete. Right?
Yeah.
And that is not necessarily the case, because people clearly annotate what they know. And nobody, so records are created while the knowledge for particular instances, for particular proteins is gathered. So at day 0 or at later days, typically this is not complete, right? So we have to somehow come up with an idea how to handle the possibility of incomplete. On top there could be mistakes in here, right? What could you do? It's pretty wild, but it actually works surprisingly. Is that why you're smiling? You have a wild idea? Or?
No, I just wish there were some reliability information too.
We're back to that.
Just like
yes,
maximum likelihood.
Nicholas, I totally agree with that. Actually, you can still do that without the reliability information.
How?
Well, you try them all. Okay. Essentially, what you do is you create all possible subvectors.
The missing part, so to speak.
Exactly.
Yeah.
And then you can simply compute for these missing subvectors something like the entropy-based classification system by some Shannon information. That's the simplest thing you can do. Now, in some sense, you may expect that this goes wrong, because you create a relatively large space and a subspace, and that this subspace really finds the right candidate. It's not apparent, but it works actually like a charm. Now, one issue here is the following that comes up. It leads, so the blue one here says, how well does that method do in terms of getting the first hit right? This is accuracy, this is coverage, so I can have a relatively high accuracy for very few targets. That means I have somehow in my prediction I have a reliability. So it is close or not close, right? The entropy has a certain value, and that value I can turn into a predicted reliability index. And that predicted reliability index, they then can say I only take predictions that are very reliable. This is the blue here. And at some point you take more predictions, so less reliable predictions, so the accuracy goes down, coverage goes up, meaning you find more things, right? That's trivial, that's the blue. The red is to actually ask, is any of the two top hits right? So blue is my first one right, in the ranking by my entropy, and red is any of the top two right? Okay, now my question to you is, red better? Is the second hit better?
Yes.
I'm asking better, right?
You have higher coverage without losing accuracy compared to the blue line.
Yes. So essentially, the way you answer that question is the red line is above the blue line at all points.
Right, yes.
Does that mean it's better?
But doesn't mean top two hits that one of the two is a hit.
Yes.
So I don't know which one is the better one, which one is the right one.
Yes, that's right.
So in the end I have 50% of, no, not 50%, but a good amount to choose the wrong one.
That is a problem, Patrick. That is a problem.
Is the correct class?
That's what Patrick says, yes.
Okay.
And since you don't absolutely, once you know the answer, you know which one. But before you know the answer, and if you always look at the first two, you have a 50-50. Totally right.
Okay.
Again, the 50-50 is a little bit skewed because you know that the top hit takes a lot here, so the blue is very close to the red. But essentially, at some point where the red really takes over, it's a 50-50. True. But my question to you now, that is true. So that may make me not very comfortable. But there's something else you have to ask here. In order to get from line is higher to better. And it is something that I talked about before. So let's just assume that we predicted two different sub-cellular localizations. Okay, the numbers cannot be right. But we have three. So we have extracellular, cytoplasmic, so inside, outside. Outside, inside, nuclear. We have three compartments, okay? Now, I can predict one of three, and I take my top two hits. If my localizations would be random, the first one would be 33%. The second one is 66%. Just random, right? One of two randomly chosen is 66% right. And when you look at the amount of increase, so the blue one clearly beats random, but the red, the increase over the blue, is not beating another random here. Okay, together they beat random because, in fact, the blue is so high. But this is an important question you have to ask. It's not only the level of, so what problems do I buy myself? Patrick is totally right, the decision making. But really, do you still? So you have to recompile random. For whatever number you look at, you have to always ask yourself, how much better than random do I do? And again, in this it's a little bit more tricky because in the red one, it's not do I do better than random. It's really do I gain over the blue enough to justify that decision of, in fact, forcing the user to get into the uncomfortable decision of, right? And in this particular case, it's actually not the case. So there's a slightly different level for different ones, but the method can in fact really find tricky relations that experts looking at these these keywords may not have recognized. So this is a little bit easy, but here you see the oxyreductase. Oxyreductase alone is absolutely not enough. And I also show you an example for the statement. So the underlying class here was larger, so the reduction of the keywords was not as simple as really if it's in all three classes. In this particular case, it violates that, but we actually had more classes. Okay, now let's get back to another idea of predicting sub-cellular localization, namely motifs. So there are two types of motifs in a protein. One type of motif is like this. So you have this on the sequence, the red signifies the motif. When you fold up the protein into a three-dimensional structure, you see the motif. When you unfold it, you see the motif. Then there is a motif of this type here, often referred to as a conformational motif, whatever, the word is not important. That is such that on the structure you see it, so there's a density of red here, a patch of red, but when you put it apart in the sequence, there's actually two patches, and they could be very far apart. So you don't easily recognize that as a common motif. Each one on its own is not only when they come together, before you know the structure, you don't have the motif, all right? So in some sense, trying to find these motifs is one of the tasks of function prediction. Now, when we get into the way in which proteins are transported through the cell, so they all start essentially on the ribosomes next to the nucleus, and from there are different decision trees here. This is a gated transport here. You go to the nucleus, yes or no, or you go into what is the secretion pathway, meaning ultimately some of these proteins, there are compartments on the way here, but ultimately some of these proteins that go on the blue path will leave the cell. Okay, and the ones that leave the cell often are marked by a signal, referred to as a signal peptide. It's called a secretory pathway. Secreted is outside of the cell, put outside. Signal peptide. So the signal is motif. Peptide is just a stretch of protein. What we know about those is they are typically N-terminal, so at the beginning of the protein. They're typically 15 to 30 residues long. They are cleaved during translocation. So what that means is, so you have a protein, that is my protein, or that is my protein. If there is a signal peptide, so say the beginning of the protein is here, then cleaved means this is cut off. I'm not going to demonstrate that now. That means that only this part of the protein is the finally functional protein. That part here is only used in order to bring it to, for instance, the surface of the protein. They do exist in all kingdoms of life, bacteria, prokaryotes, eukaryotes, and eubacteria. And they have a relatively simple, let's call it architecture. So they have a sort of beginning of the protein region, there's a hydrophobic region, there's the region where it is just before the cleavage site, and there's the cleavage site. So now you can imagine, if you have something like that, how can you use that to predict? And you see here, there are some characterizations. They have often charges. Here, the H region is at least 6 residues long. The C region is often polar and uncharged. So how could you sort of capture that to derive a relatively simple prediction method? You want to predict signal peptides? This is what I give you. Yeah?
Oh, well, you know which amino acids are polar or charged in a way. And if you have a region, well, that is something different, like unpolar and not charged, and is at least 6 residues long, then you know it's the H region.
So you can essentially define, let's call it a statistical or a potential kind of thing that looks how many polar uncharged residues do I have after these others, so I allow some spread and I compile a number, right? Let's call it a propensity. Now you could refine that by running a bunch of these, so this is eukaryotes, and sorry for the quality here, gram-negative bacteria, gram-positive bacteria. That is the cleavage site here. And what you do not see, but what I see, essentially is a letter. The height of the letter is proportional to how often you see that letter at that position. So this is an L, here is the dominant one, here you see an A, very dominant one, underneath you see a V, underneath you see a G. So essentially you can translate these counts into log-odds. These log-odds, and that's relative to the site of the cleavage, and these log-odds you can translate into some probability, right? In the simplest way you could do it like that. Then you could have a method that predicts the cleavage site. You could have that separately. That typically is a task for a machine learning device. This is what we typically would call a profile. Does that sort of remind you of any kind of machine learning device or like machine learning device? So you have a profile-like structure where you have a probability for certain states. In Markov models, ever heard of that? And that exactly is sort of one of these ideal problems for hidden Markov models. And all of these methods here go back to the groups of Gunnar van Heijnen and Søren Brunhag. So that's Stockholm and Copenhagen. And the data was collected by Henrik Nielsen. So they developed a bunch of methods that essentially, and even more, that predict signal peptides. And by now this has been so successful that there are many, many, many other methods out there. It's a relatively straightforward issue that most of the task here is to get, in fact, the right data set. And then essentially to try a bunch of different machine learning devices that do it in a biologically meaningful way. So the obvious question is, are there other types of signal peptides? So is that, in fact, so you put the signal peptide is recognized by some biological machinery, taken, protein is transported to some place, like a zip code. Is that the case in other compartments? And yes, it is, in fact. Since biology is confusing, they all use different words, but it's essentially the same thing. They're so-called transit peptides and targeting peptides. This is for chloroplast, and this is mitochondria. In principle, they are exactly the same. They're all at the beginning of the protein. They differ in the sense that the length is different, the cleavage site is different, the type of residues they use is slightly different, but essentially, they are all the same principle. You put something at the beginning of the protein that is recognized by some machinery that takes the protein and brings it, shuttles it somewhere. Okay? And another thing that they have in common, you can learn them all by simple machine learning. Okay? Or more or less simple machine learning. And then, so here really the groundwork has all been done by the groups of Søren Brunhag and Gunnar van Heijnen. Now here's a different type of motif, the so-called nuclear localization motif. That is sometimes sort of perceived more like this. So the issue here is the following. You have nuclear localization motif, NLS, often abbreviated, is recognized by proteins such as transportins or importins. They bind to this motif, bring the protein through the pore, the hole in the nuclear membrane here, import it into the nucleus, and then let go. So the importing and transporting go away. The protein does whatever it has to do in the nucleus. And sometimes there's another exporting, so protein that exports the protein here, and that recognizes the same nuclear localization signal to bring them out. Okay? It's a full circle. Now, these motifs are more complex in the sense that they are not always at the beginning, they're not always in the middle, they're not always in the end. They come at different places. Sometimes the ones that are experimentally characterized are regular expressions, and sometimes they are sort of more complex regular expressions. When we began our work, there was a database for these types of motifs, biological motifs, called ProSci. And in this, there was one nuclear localization signal, one motif, that matched in 96 proteins, and 30 of these, they represent 30 families roughly. And they then had an accuracy of 90%, meaning that they also mapped, the 10% of the proteins in which they mapped were not nuclear, or were known not to be nuclear at the time. And of all the proteins that were known to be nuclear at the time, they mapped in 3%. So what we tried to do is simply, okay, somehow you may say 3% is a small number, 90% is a fairly high number. So what we tried to do is simply do something like that. If only one exists, so let's build a couple of these. How would you do that? So find your favorite biologist and ask them. In this particular case, that didn't work. But ultimately, query the internet or papers and read what experimentalists characterized to be a nuclear localization signal. So read papers, and this is in fact just the result. Murat Çokol read hundreds of papers and extracted what experimentalists had described in these papers as nuclear localization signals. Red marks positive charges here. Those are the papers underneath, those are the proteins. And you immediately see that there's a lot of red. So essentially 2 residues, R and K, are positively charged regularly. So 2 of the 20, 10%. And you see that immediately. So one glimpse of the eye, you see this is more than 10%. Of red that you see on the whiteboard. Sometimes the motifs are a little bit more complicated, so you have several patches, or if you call that a patch. Sometimes there's really tiny density, but this one really is a localization motif. Here is more the patch idea, so you have 2 patches and a lot of X's in between. It's a little bit like that. So sometimes they are more complex. Now, some of these nuclearization signals are such that if you, in fact, somebody has done the experiment to put this peptide, this motif, onto a piece of gold, and the piece of gold is imported into the nucleus. So they work completely reliably. Okay? Now, we have a bunch of motifs. They've all been described by experimentalists. But they had their reasons to do their experiments in a certain way. So we are not fully trusting them. So how could we check them? How could we check whether they work?
Do the experiment yourself.
Yes, and here we go and have, for one of these, 3 years. So we didn't have the money, neither money nor the time for that. And actually, on top, we are computer experts. So we wanted to use the computer. Because for that, we have the money to pay for the computer.
Shouldn't they make sure that the experiment is repeatable and repeat the experiment somewhere else?
Yes, so again, repetition of an experiment is a desire that we want to have. And depending on the amount of money in the journal, this is done. In some journals, the money is just too little to repeat every single experiment in a way it's done. Under the same conditions, yes, that may not be enough. But anyway, so this is not what we can do. We cannot repeat the experiment, and this is not necessarily. We trust that they see something, but whether it's exactly that motif, we don't actually know. That has something at the level of the accuracy that the experimentalists used. They had convincing data to say that this protein was in the nucleus, right? And they had convincing data that suggested that this is the nuclear localization signal, but it may have been too short or too long. how would we find out? Or how could we check that? Remember what I showed you in one of the previous slides?
Well, if you just go for the proteins and look them up in the database,
yes.
Is the data in the database based on this, or is it because
no? So very often what happens is what would happen is that the protein here, because somebody read the paper, would annotate that as a nuclear protein, and that may be in our data set. But that still doesn't mean that the signal is in the database, right? And this is exactly the way to do it. So we have a set of nuclear proteins that we experimentally know are nuclear. We have a set of proteins that we experimentally know are not nuclear. And to sort of verify or check the motifs, what we have to do is, well, ideally we want to find motifs that only map in the nucleus. Okay? Never here, only here. Say our motif does map between the two. Can we correct that in silico? What would you do? So it matches here and matches here. What does that mean? So, for instance, this, I cannot remember whether it's a correct motif, but this could be, there could be an example for that. Maybe it matches in non-nuclear proteins. What would you do?
If it's in both, maybe believe that there was an experiment mistake, or that it actually is both in the nucleus and outside.
Yeah, that could be. That is true. But we are looking for, and these do exist, but we are looking for simple cases. We're simple-minded people, unlike you guys. No, we want to find a simple solution. And a simple solution, we're not beginning, it could well be true, what you say. But can I find a simple solution if that still helps me more than saying it could be true? I'm saying it's not true. I'm looking for the cases where it's not true. And sometimes I may find things that are the end of the line here. But what could I try? That matches the HARNT, matches in proteins that are nuclear and not nuclear. Why could that be the case? So what could you imagine that is wrong? So it may really be a nuclear protein. That is experimentally shown to be nuclear is correct. But there's something wrong with the motif. What could you try? It's not specific enough. What could you try? How could you make it more specific?
I think you said that there are some extracellular motifs.
Sorry? Did I say?
You say that there are extracellular motifs from the same protein?
So I'm not entirely sure that I quite understand your question, so let me repeat what I said. This is the motif that experimentalists have identified as nuclear localization signal. Meaning, they believe that when you see that motif, you always get a nuclear protein. However, when we look in the computer, we see that motif in a protein that is nuclear and in a protein that is not nuclear. So, by our standards, the experimentalists are not right. Nicholas is right. It could be that some proteins have both localizations. True. But let's go for some other explanation. There's something else you can try. So we have two proteins in which we map that motif. It is not specific enough. How could you make it more specific?
Look at those proteins.
Yes, and do what?
Look at the function.
Yeah, you could do all of that, but much simpler.
Maybe just a position?
What is the position?
Maybe.
Is it at the beginning or at the end? Yeah. Unfortunately, that's a great idea, Patrick, but unfortunately it doesn't help me. So let me get back to the definition of nuclear localization signals. They are really throughout. And we actually we don't see in all of this variety of nuclear localization signals we found, we did not see any rule where in the sequence they occur. Position doesn't help us. Or maybe it does. Maybe today, if we redid this and had a larger data set, maybe we would see a positional signal. But then we did not see a positional signal. So position doesn't help me. But what could we do? In a computer, again, yes, you could look at it or semi-computer, but something that it can immediately do in a computer. We did actually do that sort of semi-visually. We looked at the protein. We did exactly what you said, Matthias. But well, it's not specific enough. You want to make it longer, right? Meaning in that particular protein that is nuclear, just add the next residue. Maybe it's even a charged residue, a positively charged residue. So you extend in both directions. And see when you extend. So the first thing that you want is that it no longer matches in non-nuclear proteins. Okay? So we tried something else in this optimization. This way we sort of in silico optimized the motifs. We took essentially the core of the experimental motifs, and in some of those we refuted the motif because we actually couldn't, whatever we did, we found it in both. And then we gave up on that motif, and it could be a true signal. Okay? But then it's not a motif in the sense that it clearly shows when I see the motif, I know nuclear. Right? So for us in that sense it didn't work. But there is an additional thing that we try to do, is that we try to have the motif map into non-trivially related nuclear families. Meaning that if I have two proteins that are nuclear and they are 100% or 90% sequence identity, and I map in those two, that is not enough. I want to map the motif into things that I cannot recognize as homologs. And only then do I say this is really an informative motif. With this I throw out a lot of motifs, but the ones I have, in fact, are optimized such that they help me a lot. Ultimately, at the end of the day, this created a data set of 214 motifs. So we went from 1 to 214 after a few months. And overall, by design, at the database that we had then, they were 100% right. So we chose them such that they only mapped to the nuclear proteins, so that they never mapped to anything we at the time knew was non-nuclear. So this is by construction. And then they mapped sort of 40% of all the then non-nuclear proteins. And it was a relatively simple project. In some of the cases where we mapped it, we saw that this was in fact what you see highlighted here is the nuclear localization signal. That you may recognize as the double helix, the DNA. So this protein really binds the DNA, and you see that it binds the DNA exactly at the nuclear localization motif. So you see exactly this cycle. Importing recognizes the nuclear localization signal. Importing goes off. The same signal binds to DNA.